Hello everyone. Welcome to the 2025 UFAW International Animal Welfare Conference. My name is Huw Golledge and I'm Chief Executive of UFAW. It's an absolute pleasure to welcome over 500 of you from more than 60 countries to this conference this year. It's absolutely fantastic to see the number of people who register for these meetings every year and to see them growing. We're absolutely delighted that so many people are engaged with animal welfare science. So once again, welcome. If you're following this meeting on social media and posting about it, please tag us or mention us in your posts. We'd love to see what you're talking about as the meeting progresses. The hashtag is UFAW 2025, and we'll be posting on LinkedIn and Blue Sky, but we are aware that there are other platforms. So if you're posting on there, we'll keep an eye on those as well. Before we get on with the main focus of the meeting, I always like to just give a bit of an introduction to UFAW and what we do, because I'm aware that lots of you are new to this conference and new to UFAW. So UFAW is a charity. It was founded almost a century ago, and for all that time, our mission has essentially been to promote science in the service of animal welfare. In other words, we try to discover what matters to animals, develop scientific solutions to animal welfare problems, and disseminate evidence-based animal welfare information, all with the hope that that improves the welfare of animals. And you can obviously read more about what we do on our website, ufaw.org.uk, but I'll just highlight a few things that are hopefully of interest to people attending this meeting. In terms of our mission to discover what matters to animals and develop solutions for those problems, largely we tackle that by trying to support scientific research, either by funding that research where we can through grants, or by supporting career development for people who do animal welfare science, and in particular trying to generate and develop the next generation of animal welfare scientists. And I'll explain a bit more about how we do that in a couple of slides. In terms of disseminating the evidence about animal welfare, obviously this conference is a key way we do that, as are our other meetings, but we also publish our scientific journal, Animal Welfare. We have a book series, and we publish lots of information on our website. So thinking about research grants, we have a grant scheme that's open for anyone to apply to at ufaw.org.uk/grants. We fund a range of projects, lots of quite small projects, and occasionally when we have the funding, larger, up to six-figure projects. And you can see some of the examples of the things we fund on our website. Some of our smallest grants are student scholarships, which fund short, several week projects, typically for undergrad or sometimes master's students, to undertake as their first bit of animal welfare research. And we think those are some of the most impactful things that we fund. Lots of people who had their first exposure to animal welfare science doing one of those projects have gone on to either make a career in animal welfare research or in another area of animal welfare. And we're really proud of that scheme. So do have a look at both of those if they're of interest to you. Another thing we do, which is really crucial, is that for students who are undertaking those early projects at the start of their career, we have a mentoring scheme where we try to provide some support, particularly for students who are really keen to do this kind of research, but perhaps come from an institution that has less of a history of doing animal welfare research, and where they may find it difficult to get the support to put a high quality application for funding together. So what we do is pair those students up with early career researchers, typically at the sort of postdoctoral stage of their career, to get them some support and mentoring. And this has been a really successful scheme. It works really well for the students who are mentored, but it's also proved really valuable for the early career researchers who act as mentors. It's often their first experience of supervising or helping to supervise a student. And I think everyone involved in the scheme finds it really rewarding as well as beneficial. So again, if you'd like to get involved with that, either as a student or as a mentor, please do have a look at the link. UFAW is an international society, and we're incredibly keen that although we're based in a little office in the UK, our influence and our support is spread around the world. And one of the best ways we do that is through our Links scheme, where people represent animal welfare science and UFAW at universities which have an active animal welfare research or teaching program. So we provide a small amount of support for local animal welfare science activities like meetings or student clubs and so on, and also share our information with those universities in the scheme. And if you're at a university that isn't part of the scheme, please do consider signing up, particularly for anyone who's in parts of the world where we're less well represented at the moment. I mentioned our journal, Animal Welfare. It's the longest running continuously published animal welfare journal, founded in 1992. In 2023, it went gold open access, which means anyone can read anything we've published since then, so there are no barriers to access to the papers we publish anymore. And although we do charge an article processing charge, we have a really extensive waiver scheme for anyone who can't pay article processing charges. That's a blanket scheme for a large number of low and middle income countries, and for anyone outside of those countries, they can simply apply for a waiver if they don't have grant funds or other funds to support publication. So there really are no barriers other than the quality of the research to publishing in animal welfare anymore. We've recently launched collections in the journal, which are groupings of papers on a specific topic, and we've launched a number of collections. The most recent one is on positive animal welfare. And the advantage of these is that, as well as the more recent papers, all the historical papers which we did publish before we went open access under that theme are all available open access now. So do have a look at those collections if that's of interest. The editors for the journal are hopefully well known to you: Dr. Beate Nielsen, who is a research director at UFAW, and Bas Rodenberg, who lots of you will know, and our managing editor, Steve Weddell. Also, lots of you who've published with the journal will know. Steve's a vet. What this means is everyone you deal with at the journal is an animal welfare expert, and we think that gives you a really excellent service when it comes to publishing your paper. Unlike some other journals, everyone you deal with will understand what you're trying to publish, they'll understand the message you're trying to get over, and they'll be working to help you publish excellent research. You can follow the journal on social media. We're on LinkedIn and Blue Sky. We publicize every paper we publish. So that's a great way to follow what's going on with the journal. Just to promote a couple of other UFAW events that are coming up. A really interesting one that we launched this year with our friends at the Animal Welfare Research Network is our seminar series on translating animal welfare research into law and policy. And this came out of concerns that animal welfare scientists do lots of amazing research which policymakers should be taking notice of, but they're perhaps not the best at bringing it to the attention of those policymakers. So what we've been doing is having a series of seminars from people involved on the policy side, explaining how the policy making and legislative processes work and how scientists should engage to bring animal welfare science to the notice of policymakers. We've had a few in the series already. The next one is next week from George Eustice, who's a former Secretary of State for Agriculture in the UK. So we'll be getting a sort of insider's view from the top of government about how to engage with the policy process. And then there are several more seminars in this series scheduled in the autumn. If you register, you can also have access to the recordings of the previous seminars, and that's all free to sign up to. So I'd urge anyone who wants to try and increase the impact of their research to have a look at those. Next year will be UFAW's centenary. Amazingly, we've been around for 100 years next year. We were founded at the University of London in 1926, and we're planning to return to the University of London to host our biggest ever conference next summer, and we'll share full details of that very soon. But I hope to see you there next year, either in person, or we'll make sure that it's a hybrid event for those of you who can't attend in person. So that's pretty much my whistle-stop tour of some of the main things UFAW does. If you value that work, we'd love you to engage with us if you're not already. The best thing you can do is become a UFAW member, which both supports our work and offers benefits to you, like reduced conference registrations, discounts on some of our publishing, book publishing and journal publishing, and so on. So please do consider becoming a member. If you are already a member or you don't want to sign up as a member, at least stay in touch by signing up for our updates or following us on social media. And if you would like to support us even more, bear in mind UFAW is a charity. Absolutely everything we do only happens because people donate money to support our work. So we would be really grateful for any support to continue doing work like this meeting. So on to the actual conference, now that I've given you an amazing sales pitch for UFAW. Just to outline the program, we've got 3 days of really exciting scientific talks coming up, starting today at 1 o'clock, as we just have. But to note, the next 2 days start at different times. Tomorrow starts at 10 a.m. GMT +1, which is British summertime, and Thursday starts at 8 a.m. And the reason we do this is to allow the speakers in particular and also the attendees to attend at times which are easier for their time zone, so that we don't have to have people speaking in the middle of the night or at the crack of dawn. So apologies if that's slightly confusing with the start times, but they're different every day for a reason. And what you do is every day or every session of the meeting, you log on with the same link, the one that you've just logged on today. The program is available at ufaw.org.uk/program25 if you don't have it, and it's also available in the attachments folder on GoToWebinar. Just to let you know, the sessions are all being recorded, and as soon as we've processed them, they'll be available for registered delegates to view after the meeting. So we'll share those links with you once they're ready. Hopefully you're looking at a toolbar like the one I've got on this slide here in GoToWebinar. If you could keep your mics and camera off just so that we don't have any interruptions for the speakers, that would be really helpful. If you have questions for the speakers, and we encourage you to ask questions about all the talks, if you click on the little question mark icon, you can type a question in the box and we'll select questions for the speakers. If you've got any technical problems, you can also put those in there and we'll try and help with those. And next to that is the attachment icon with a little paperclip on, where you can find a copy of the abstract book if you need that. We also have a virtual poster session for this meeting. We'd love you to visit the posters online and ask questions, ask written questions of the posters, and also vote for your favorite poster in the poster competition if you follow the link on the slide now. So please, in the breaks or after the end of the meeting, please go and have a look at the posters. So that brings me to the end, except to thank our headline sponsor for this event, who are Charles River. They've generously supported this event and enabled attendees who would have otherwise struggled to pay the registration fee to attend for free. So thank you very much to Charles River for their generous support. So after that long preamble, we get on to the part that everyone's been waiting for, which is the first scientific session. And I'm afraid you have to put up with me for a little bit longer as the chair of this session, but I will hand over to other chairs who've generously volunteered to chair after this one. So I will stop sharing my slides at that point and introduce our first talk, which comes from Olga Burenkova. and she's going to be talking about how does improving welfare improve cognition, investigating the impacts of housing conditions on neural markers in zebrafish.
Hello everyone, my name is Olga Burenkova. I work at the University of Guelph, Canada, which I gratefully acknowledge as a part of the traditional territory of the Mississaugas of the Credit First Nation. It's a great pleasure and a privilege to be speaking at this conference today. In this talk, I'll discuss how improving animal welfare can enhance cognitive function by exploring the impact of housing conditions on neural markers in zebrafish. Zebrafish are small species of freshwater fish originally from Southern Asia. They are a very popular model organism with at least 5 million used in research per year worldwide, particularly in studies of developmental biology, neuroscience, genetics, and toxicology. In a lab setting, zebrafish are typically housed in an empty tank such as this one, where their natural habitat includes vegetation and sediment in shallow, slow-moving streams. Dr. Michelle Lavery, working on her PhD in Dr. Mason's lab, showed previously that zebrafish actually prefer some components that mimic their natural environment. She split zebrafish tanks between barren conditions and those offering naturalistic resources such as plants, gravel, or their combination, resulting in 4 conditions. Then she measured the amount of time they spent on each side of the tank until they stabilized over a number of days. In these settings, fish were found to prefer the resource conditions in a rank order, this with plants plus gravel more preferred than gravel alone, which was more preferred than plants alone, which was more preferred than the empty tank. These short-term preferences, expressed over days to weeks, did appear to have long-term welfare implications, since compared to fish housed for months in the most preferred conditions, those housed in the least preferred barren shows 2 signs of chronic stress: elevated mortality and reduced attractiveness to the opposite sex. But to neuroscientists, so-called enrichment, a term we tend to avoid as we feel preferred resources are essential for animals, is not about welfare, it's about cognition. In the next part of her experiment, after many months of rearing in these 4 groups, Michelle used the T-maze task pictured here. Fish had learned to associate one arm of the maze with a reward by swimming to that arm to receive food. The fish that learned the task the fastest were those from the most preferred housing conditions, grass plus gravel, followed by fish from other conditions in descending order of housing preference. This suggests that improving welfare improves cognition. But how? What was going on in the brain of this fish that led to these differences in cognition? That's what we are going to test here. The aim of our study was to investigate the effects of long-term housing in 2 contrasting environments, either the least preferred barren or the most preferred, with plants and gravel, on key neurobiological markers of neuroplasticity chosen primarily from rodent enrichment work and fish rearing studies. The first 2 markers are measures of neural proliferation, brain cell density obtained using the nuclear marker DAPI, and density of cells containing proliferating cell nuclear antigen, BCNA, a marker for 2 opposing biological processes: cell proliferation, new cell formation, and cell reparation. Levels of brain-derived neurotrophic factor, BDNF, a protein supporting neuronal differentiation, survival, and growth, as well as synaptic function. And then level of acetylation of lysin 9 on histone H3, an epigenetic marker associated with higher gene activity that might support synaptic plasticity and learning. In mammals, studies by Michael Meany and other colleagues from the field of behavioral epigenetics showed that stimulating early life experience like high level of maternal care can shape brain gene expression through epigenetic changes such as histone acetylation, and this influences stress responses and brain function expressed in better learning. Yet in fish, the role of histone acetylation response to environmental enrichment remains unstudied, making it a real promising direction to explore how experience shapes the brain at the molecular level in non-mammalian species. 40 brains were collected from zebrafish previously kept in either barren or the most well-resourced tanks containing gravel and grass, living there for 6 months, 20 animals, or 20 months, also 20 animals, half male, half female, starting from the age of 3 months. Then we sliced the brains into thin sections, stained them using immunohistochemistry, and took images under a microscope. They were further processed in ImageJ to extract brain markers measures and statistically analyzed by linear mixed modeling. You can see here a scheme of zebrafish brain, which is tiny, about the size of a small grain of rice, but still highly differentiated into regions. Brain markers were assessed blind to housing in 3 of these regions: the dorsolateral and dorsomedial telencephalon and the optic tectum, all regions shown to be stress or enrichment sensitive in fish studies and also involved in learning. In this image you can see examples of zebrafish telencephalon sections stained for the 4 markers we used. The yellow boxes indicate the counting regions for the lateral telencephalon, while the blue boxes correspond to medial ones. The insets show magnified views of the markers. All labels are localized within the cell nuclei, except for BDNF, which is localized outside the nuclei. No structural differences assessed by brain cell density based on DAPI staining were observed across any brain regions, indicating its overall stability. PCNA marker of new cell formation and cell repair was unexpectedly decreased in the dorsolateral telencephalon after housing in well-resourced tanks, and in the optic tectum this marker still remains to be analyzed. A level of neurotrophic signaling, BDNF, appears to be unaffected in the telencephalon and is barely detectable in the optic tectum due to very low signal. Histone acetylation was increased selectively in the optic tectum of fish from well-resourced tanks. Regarding the demographics, BDNF and histone acetylation level were increased in females. Age was associated with the most consistent decline across markers, particularly affecting proliferation, PCNA, neurotrophic signaling, BDNF, and epigenetic regulation, histone acetylation. Here you can see the graphs with these differences: lower level of proliferation in telencephalon of fish from well-resourced tanks, but higher level of histone acetylation in their optic tectum. Turning to conclusions, well-resourced fish had significantly higher histone acetylation levels in the optic tectum, suggesting enhanced transcriptional activity and synaptic plasticity. These molecular changes are consistent with findings in mammals. Such epigenetic modulation is one of the pathways through which improving animal welfare by providing stimulating enrichment environments can support cognitive function by promoting brain plasticity. If this reflects the mechanism that enhance learning abilities, in the following 4 group analysis, we should see slightly elevated levels in plants-only fish and higher levels in gravel-only fish, and the maximum level of histone acetylation in the most well-resourced subjects. Interestingly, contrary to our expectations, well-resourced fish had a lower PCNA positive cell density in the dorsolateral telencephalon. This is quite puzzling result at the first glance, but considering that increased PCNA levels can also indicate cell repair in response to damage or stress-induced cell death, the elevated density in barren fish may reflect a compensatory response to poor conditions rather than enhanced neurogenesis. PCNA data from the optic tectum are still pending, and it will be exciting to see how this another proliferative niche responds to differential housing. Also surprisingly, that BDNF levels showed no significant differences across groups, despite its well-established role in neuroplasticity. Volume measurements of regions of our interest may still reveal housing effects. To conclude, our findings underscore the complexity of brain plasticity and its sensitivity and also non-sensitivity to environmental conditions. Perhaps our enrichment conditions weren't strong enough to elicit clear effects. But this is where we are. So wish us luck as we continue to investigate the central question: how does improving welfare improve cognition? I would like to say sincere thanks to my colleagues for their generous support, productive collaboration, and valuable insights that shape this work. Thanks for your attention, and I'm looking forward to questions.
Sorry, we cut you ever so slightly short there, Olga, but thank you for a wonderful talk. There's lots of questions coming in through the chat now, so I will go straight to some of those. So, 2 seconds. First of all, from Lindsay Kitchenham, why do you think zebrafish brains' responses to enrichment differ from those seen in mammals?
Thank you for your questions. First of all, I would say that the fundamental mechanisms of neuroplasticity between mammals and fish, I would say they're pretty similar. They are pretty fundamental between species. But while there are still some minor or some details with the detailed differences, for example, neuronal proliferation in zebrafish, particularly in fish in general, is much more profound than in mammals in terms of rate of proliferation, regions specific having this proliferation zone, and other aspects of proliferation. So in general, the mechanisms are pretty similar, pretty fundamental, with minor differences. But I would say that the results, what we have right now for this project, they are displayed not mainly differences with mammals, but even differences with existing literature shown in fish. I would say not very abundant corpus of literature. For example, the only paper already, the only study conducted already in fish studying proliferation in response to environmental enrichment or exposure to well resource cages, tanks, they show increase in proliferation. But I would say that the main differences what we see right now, it's maybe protocol of exposure of animals with our environmental differentiated environment, because in this previous work they only expose fish with 1 week of environmental enrichment, so they had very acute effects of the brain response to this acute exposure, while as you have seen in my presentation, we had multiple months of exposure. So I think the differences will be not mainly about the species or taxa, but mainly about protocols of exposure. Thank you so much.
Thank you. Thank you. I've got one from Paul Rose here, who wants to know if the plants used were plastic, and if so, if real plants were used, would that create a more dynamic environment that would further benefit animal welfare?
Could you please repeat? I got the material. Yes, indeed, material was plastic. And the second part of the question, could you please repeat it?
He's wondering if you used real plants instead of plastic, whether that would have a larger welfare benefit, because it would be a more dynamic environment, as he puts it.
Of course, yeah, of course. It's almost trade-off, because the plastic plants, of course, they are less naturalistic, but at the same time they are more standardized, because we can expect that the real plants could be mainly in different conditions, maybe health conditions, plant health conditions. We know that they also can decompose, etc. So it's much harder to standardize, I would say, them. But beneficial, of course, it will be more beneficial.
Yeah. Like, definitely
in terms of not for researcher, it will be less beneficial maybe, but for fish it will be more beneficial. Yeah, so kind of trade balance. Yeah.
So I suppose a follow-up on that, is there any halfway house where you can create a more realistic enrichment that might be even, might more closely mimic a living plant? I don't know.
Maybe. I don't know.
Yeah, no worries. I guess kind of related to that, as a question from my colleague Steve Wickens, who's asking how likely you think it is that people will change the way they house their fish based on your findings.
I think that for researchers in this area would be more like, in terms of welfare, more maybe important finding will be like general finding from, not my part of project per se with brain markers, but from general finding that the mortality of zebrafish who were grown and who lived in these barren conditions are much higher mortality level than in well-resourced tanks. I think this is really important, and this deserves to like give a hint that it should be alter the way how we actually grow our fishes. But for fundamental researchers targeted with whose attention is targeted on brain markers, I think it's important stepping stone to one of them, like stepping stones to study how exactly housing conditions are related with improvement of cognition, what are potential mechanisms. And of course it's very like initial studies with no distinctive answers, and a lot of steps are still before us. But anyways.
Yeah. But I think you raise a good point, and it's the fact that if the mechanism is understood as well as the beneficial effect, that maybe makes an even stronger argument for the researchers. So I'll go for one last question from the list. So this is from Jonathan Amory, who says, I remember a study where pigs raised in barren environments were faster in mazes compared with those raised in enriched conditions, and that was suggested to be about differing motivation rather than learning. Is that possible here?
Here, unfortunately, I do not know this work who is cited on pigs, and it's of course it's really depends on. I totally agree that what protocol we use, what kind of motivation, is it kind of negative motivation or reinforcement or positive reinforcement, and it really depends on the situation. So in our protocol of learning, we used positive reinforcement, and it like worked pretty well. But I would generally assume that these it's more affects learning, like cognitive. Would be my first choice. It's cognitive abilities of animals, which is not. It's not alternative either this or this. I would say that it easily could be the mixture of these two effects, which is sometimes really hard to distinguish between them.
Okay. Brilliant. Thank you very much. So thank you for an excellent presentation. There's more questions in the question thread that we might be able to answer later. But thank you so much.
Thank you.
So thank you very much, Olga. I'm going to move on to our second talk of the day. And this one comes from Cynthia Schuck-Paim and is entitled The Pain Echo Chamber Effect, How Cages and Barren...
Huw, that's the wrong one. That's the third talk.
Not on my list, Steve.
It's Katrina.
Okay, which one am I doing?
Yeah, it's Katrina next. Affect pair housing on dairy calf welfare.
Sorry, I apologise to everyone. Then we've moved those around. So first up is Katarína Bučková with the effect of pair housing on dairy calf welfare, a systematic review. Apologies.
Good afternoon or good morning, and thank you that you attended this talk, and also UFAW for inviting me here as a speaker. I'm more than happy to present the results of our systematic review on effect of pair housing on dairy calf welfare. You may be wondering why we decided to write a review on pair housing for dairy calves. So recently there has been an increase in alternative housing systems for dairy calves, and this could be illustrated by using the UK as an example. In this country, the use of individual pens for dairy calves decreased significantly over 12 years. Therefore, everybody who is involved, for example, dairy producers, animal welfare researchers, or policymakers, need accurate information on alternative housing systems for dairy calves. However, there was no review on pair housing, although there has been extensive research. The only similar review was written by Costa et al., but the authors didn't make any specific conclusions or implications for pair housing, which was reviewed within group housing. We would argue that pair housing doesn't have to necessarily provide the same benefits, and it can also face different challenges than group housing. For example, we could expect that health of pair house cows would be better because they are only housed with one animal. So the objective of this study was to summarize and critically evaluate the current knowledge on pair housing of dairy cows compared to individual housing. Briefly, our material and methods. We search for databases using the keywords you can see under the first bullet point. After literature search was done, duplicates were removed and inclusion and exclusion criteria were applied. We only included peer-reviewed full-text primary research articles written in English, and they had to contain control individual housing. At the end, there were 36 articles left. We read the results of these articles in full, and they were categorized into 6 categories. Now, I would like to present our findings. First of all, grow and feeding. We found that pair housing either improve, grow, or concentrate intake in calves, or there was no difference between housing treatments. If positive effect of pair housing was found, this was mostly the case of cows that were fed higher quantities of milk, meaning at least 8 liters per day, which is higher than the current commercial practice. Surprisingly, there was a very limited number of studies on other feed-related variables such as hay intake, water intake, or feeding behavior, so we can't really make any conclusions on these. Now let's discuss health. As you can see, there were various health parameters assessed in calves. Mostly it was diarrhea and respiratory problems. In general, we found no effect of housing treatment on calf health. However, I can't make any conclusions on specific variables because often there were only a few studies, for example, on pathogens in feces or development of antibodies. If there were not many studies, so there could be increased risk that health is affected by confounding variables such as calf age or quantity or quality of colostrum fat. Now let's discuss coping with stress. Again, you can see that there were various variables assessed, but in general we can say that pair-house cows coped with stress better. And this could be due to social buffering effect, when cows housed in pairs benefited from social presence of familiar conspecific. The only exception was human approach test, and here we found that individually house cows were actually more motivated to approach humans. This could be because they were socially isolated from other cows, so they switched their attention to people, and also they could be more bored. Again, you can see that on some variables we didn't have many studies, and sometimes we only found one study, for example, on transport or disbalance, so I'm not going to make any specific conclusions on these. Another issue we faced was that studies on novelty mostly used novel arena and or novel object test, and these tests have been criticized because they may lack scientific validity. For example, if social animals like calves are tested alone in a novel arena, so we can't be really sure what we are measuring, right? Because we could be measuring their reaction to separation, from conspecific to novelty, or to both. In case of novel object, there were issues related to methodologies. Researchers sometimes tested cows in home pen, sometimes in a novel arena, and sometimes in the arena which was familiar to calves. Also, different novel object was used. Okay, let's discuss behavior now. We categorized behaviors into three categories: activity, social behaviors, and abnormal or stereotypical behaviors. We didn't find any differences between housing treatments in activity, but we found that per-house cows seem to have better developed social behaviors. The results on abnormal or stereotypical behaviors were mixed. Again, there were some issues. For example, this is really simplified because there were various behaviors in each category. In activity, for example, we had walking, standing, lying down, use of space, and the question here would be how to interpret if there was difference. Unfortunately, we can't make any conclusions on emotional states because here we only had 4 studies, one on cognitive judgment bias test and the other 3 on play. When it comes to learning abilities, the results were different and methodologies also differed, for example, in age when cows were tested or in tests used. Now I would like to discuss more general study limitations and future directions. I already mentioned that we had issues with limited number of studies and interpretation. Besides that, we based our interpretation of the results on statistical significance. This would mean that if authors reported trend, we would still say there was no difference between individually and per house cows. Now, future directions. So I already mentioned some important variables relevant for cow welfare were almost absent, only 3 studies on play, and I already mentioned that besides feed intake and growth, there were only few studies on other feed-related behaviors. Researchers mostly assessed parameters in cows before and shortly after weaning. This is of course reasonable because weaning from milk is very stressful for cows, but we still think that it will be valuable to look how animals are doing later in their lives. And please take our results and conclusions with caution because our study is still going through interactive peer review process. So your tentative take-home message is that pair housing may improve concentrate intake, growth, social behavior, and coping with stress in dairy calves. However, individually housed calves seem to be more motivated to interact with humans. We are not quite sure if this is bad or good for animals as well as for personnel. It could be advantage as well as disadvantage. For example, if dairy cow would like to play with farm stuff, it could be unsafe, but it is more time consuming to work with two shy animals. And we didn't find any differences between individually and pair house cows when it comes to their health. Last but not the least, I would like to acknowledge our funders. Again, I would like to thank UFAW for this invitation and you for your attention, and I hope I will get some great questions. Thank you.
So thank you, Katarína. Welcome. I'm going to take the chair's privilege first, and I confess I haven't looked at your preprint, but have you made any recommendations about how the quality of some of these studies might be improved in your paper? Because you've highlighted some really important methodological issues, I think.
Yes, we did some, but actually, yeah, there is certainly space to do so. Like sometimes some variables were not reported, like calf age. So we basically didn't know like what age calf was tested at, and sometimes it was the same for some other variables, like let's say like how many animals were in group or something like that. And the issue which I mentioned at the beginning was that most studies were based on p-values, right? So that's the way we chose this approach, because otherwise it would have to be really meta-analysis. So maybe we should adopt more Bayesian statistic, like to also look at effect size and so on.
Fantastic. Yes, I think that's really excellent to point that out. I'm going to go on to the questions which are all appearing rapidly in the question thread. There's one from my colleague, Beate Nielsen. If pair-housed calves were fed more milk, could this be the cause of differences found rather than the housing?
Yeah, so definitely we saw that trend, that if they were fed more milk, so there was almost always difference. So yeah, definitely it could be like important confounding variable, I would say. So yeah, so that's why I said almost everywhere like it must be interpreted with caution, because there were so many confounding variables, and one of them was amount of milk fed. Yeah.
Excellent, thank you. And another one from Jessica Kate, who says, thank you for the presentation. I was wondering if you considered conducting a meta-analysis of the studies. It seems like some were interpreted as showing neutral effects, but I'm curious whether a meta-analysis might give a clearer directional effect overall.
Yeah, like it was hard to choose if we are going to do systematic review or meta-analysis. Then finally we opted for systematic review, but during peer review process, we actually got a recommendation from one of reviewers to conduct meta-analysis. But meanwhile, another meta-analysis on this topic was published. So we just stick to systematic review at this point, and they also considered group size. So they also look like if it differs, if there is only one calf as a companion, on if there are more calves in the group. So basically, this has been recently done. It's a really recent meta-analysis, I believe. Like it was published last year or something like that. Yeah. So.
Okay, makes sense. Thanks. One from Beth McKeague. I think you've answered half of this, but maybe not the other half. So she says, great talk. What are your hopes for the application of this review to either research or industry? So I guess particularly, like, is there anything that the industry can take from this?
Yeah, so what was our main goal was that really producers know what to expect when we house two cows together, because sometimes they are really worried about some negative effects, like that cows in pairs could get sick more often because they are housed with another calf, and also sometimes they believe that they grow worse. So our aim was actually uh, to look at if these are true or not, um, and now we can say, okay, so if you house 20 cows together, so based on other studies, of course not that, because this was not our aim. So we can say with 20 cows you can maybe have increased disease transmission, but we really show like if you house two cows together, so you shouldn't be worried about health. So yeah, so we would definitely like to see, yes, that finally some beliefs are overcame.
Thank you. One from Misha Kaluza, who says their question is about the appropriate time with starting the pair housing system after calving. And they say they ask, especially because of the key steps like intake of colostrum and starter in the first day of life.
Yeah, so opinions on this differ a little bit, because somebody would say like it's okay, it's almost okay if they are housed immediately together, but mostly we should wait approximately 1 week to see how calf is doing in general, like maybe weaker and really sickish animals should be housed individually. Yeah, so most common recommendation is to wait 1 week and then pair them.
Thank you. A quick technical question from Nicole Basti. Were the studies carried out on commercial farms or research farms?
Yeah, so mostly it was a research facility, right? And it was always single farm. So something what we would like to see is maybe use animals from at least two different farms. But really, this was a rule. There was always a single farm, and it was mostly research facility. And in some cases, but this was less common, it was a commercial farm.
Okay, thank you. And one last one, again from Beata Nielsen. Is the reason that more milk is fed to the pair of calves just to ensure that each gets enough, or is there another reason for the fact they
Yes,
get more
so
milk?
I maybe said it wrong. So all cows, because there are always two treatments. So we only included studies with control, individual housing, and with per house cows. And in these studies, cows were always fed the same amount of milk. But if if per house cows, sorry, cows growth was improved, so in this case, all these cows in the study got more milk.
Okay, that makes sense. Thank you very much. Okay, brilliant. Well, thank you for answering all those questions, and thank you for an excellent, really clear talk as well. Thank you very much.
Thank you. Have a good one.
So moving on to the talk that I've already introduced once, and it's definitely this one this time. So I'd like to introduce Cynthia Schuck-Paim, who's presenting the pain echo chamber effect, how cages and barren environments may amplify pain perception in animals.
Today I want to talk about pain, not as a symptom to be managed, but as an experience that is very much shaped by the environment where it happens. Look at this injury. As welfare scientists, as veterinarians, we are trained to assess it, to score it. We have models to assign a severity score to this exact condition, to understand the impact of this condition on the welfare of the animal. But I want to ask you a question. Assuming this footpad injury has exactly the same size and severity in both these environments. Is pain the same for a chicken foraging in a complex, more natural environment as the one on the left, as it is for a chicken in a barren battery cage as the one on the right? So if we were to treat this injury, should we use the same type and dose of analgesic? What I want to discuss today is that pain is also a product of place. We have long treated pain as a relatively fixed response to a specific injury. So the assumption is that a fracture is a fracture, a lesion is a lesion. And today I'm here to argue that this assumption is not only wrong, but it leads us to systematically underestimate animal pain in most intensive production environments. The idea is that the environment is not passive. It's also one of the most powerful modulators of pain that there is. To understand the impact of the environment on pain perception, we must first look at the specific biological mechanisms that animals have to regulate pain, and I'll start with pain suppression. One of these mechanisms is given by what is known as gait control theory. For example, when you have behavior engagement with movement, with touch, when an animal is walking, exploring the environment, foraging, it activates large, fast nerve fibers that detect touch, movement, vibration. So according to gait control theories, these signals reach the spinal cord relatively fastly and trigger inhibitory neurons that literally close the gate on slower pain signals trying to get to the brain. So the outcome is a relatively powerful immediate analgesic effect. So that's one of the mechanisms. Another mechanism of endogenous analgesia is the release of the body's own pain-relieving drugs. So, for example, when an animal is engaged in highly motivated behaviors like nesting or active foraging, exploration of the environment, it stimulates the brain primaries descending, let's say, pain control circuits that is different in different groups. It's different for vertebrates and fish and mammals and birds, but it's there. And this pathway releases the body's own natural opioids, for example, endorphins, which have the power to block pain-promoting neurotransmitters. So this is a very powerful opioid-mediated analgesia that can partially, or in some cases, completely suppress pain behaviors and block pain signals. It's why in all the experiments in the 90s, for example, you saw complete pain suppression in chickens engaged in the exploration of novel environments or in nesting behavior. Social contact, proper social contact, like the presence of familiar individuals, particularly affiliative touch, triggers the release of oxytocin. And oxytocin also has its own analgesic, anxiety reducing effects, and it acts on the brain to reduce both the sensory qualities and the negative emotional qualities of pain, and it creates a sort of social buffer from pain as well. Finally, there is the simple power of distraction through cognitive load. So the brain has a limited capacity for attention. When an animal is engaged in a complex task like solving a puzzle or navigating a new environment, its attentional resources are partially or completely consumed, and this creates competition in the brain. So the brain literally has less capacity available to process incoming pain signals. The pain isn't gone, but its perception is significantly blunted because there is not enough attention for the processing of these pain signals. That's why in some countries, lower income countries, where there isn't enough resources for painkillers, distraction is commonly used as a pain mitigating strategy. Those were some of the tools in the body's natural toolkit for pain suppression and recovery. Now I'll discuss some of the mechanisms that actively amplify pain, increasing both its intensity, perceived intensity, and duration. Okay, chronic stress. I think this one is not surprising. When there's long-term, inescapable stress, the body is flooded with cortisol until its receptors become resistant. So this removes cortisol's natural anti-inflammatory effect and leads to the opposite result. So the brain immune cells become primed and adopt a pro-inflammatory state. When there is any painful stimulus in that state, the body overreacts by releasing a flood of cytokines. There are substances that will lead to an amplification of the pain signal, leading to what is known as central sensitization. So the animal will be in a state of hyperalgesia, so an extreme or exaggerated reaction to pain signals, perceiving the intensity of the signals as higher than they would normally be. Okay, so chronic stress also have other effects that will affect the duration of healing and duration, consequently, of pain. So in addition to the effects I mentioned before, there's also three other effects that are a result of this sustained levels of glucocorticoids in the body. The first on the left is immune suppression. So the glucocorticoids inhibit the function of key immune cells and compromise the ability of the body, for example, to clear pathogens, here on the left. Then there is the inflammatory dysregulation. Effective healing requires a well-timed transition between a pro-inflammatory phase, the initial pro-inflammatory phase, and a restorative phase, anti-inflammatory phase. And chronic stress disrupts this transition, prolonging the inflammatory phase. So it delays the resolution of inflammation as well. And finally, there is the inhibition of cellular repair. So, for example, cortisol inhibits the activity of fibroblasts, which are essential for tissue reconstruction. So wound healing is delayed in the end. So as a consequence, wound healing is delayed, you feel pain for more time, or it takes longer for pathogens to be cleared, so infections take longer to heal as well. Common factors underlying chronic stress: high stocking densities, unstable social environments, either social isolation or unstable social groups, chronic behavioral deprivation of motivated behaviors, lack of movement, confinement, poor air quality, poor water quality in the case of aquatic animals, disrupted rest, disrupted sleep, noise, vibration, improper lighting, chronic feed deprivation, which is common for female breeders, and so on. Another mechanism of pain amplification, so the amplification of the perceived intensity of pain, is pain itself. So unrelieved pain initiates this process of central sensitization that can happen in two ways. One is a functional way, a phenomenon known as temporal summation. So when there are repeated pain signals arriving at the spinal neurons, each pain signal received may produce a larger response than the one before, progressively amplifying the pain message that is sent to the brain. So over time, this, let's say, hyperexcitability can lead also to the physical rewiring of the circuits themselves. So this is what you see here in the figure. On the left, you see a normal circuitry where you have, for example, nerve touch sensing fibers leading to the leading to the brain and not causing any pain. But when you have a rewiring because of constant pain, for example, new branches can grow and sprout into the pain circuits. And in this way, normal sensations like touch, like movement, may lead to pain. This is a state called allodynia, in which non-painful signals may lead to pain given this physical rewiring of the circuitry of the brain. And finally, a well-documented mechanism is adversity early in life. So any adversity, pain, or stress early in life can completely change the development of the nervous system. So painful procedures or even maternal stress during gestation can cause epigenetic changes and modify how genes are expressed without changing the DNA itself. So these changes can switch on pain receptor genes and switch off or turn the volume on the genes for the natural opioid system. What it means that the animal is essentially programmed for a life of heightened pain sensitivity. This phenomenon has been very well documented in pigs, for example, in chickens, in mammals, and where you see, for example, offspring of stressed mothers or mothers that were stressed during gestation, the offspring having increased pain sensitivity. Also, animals exposed to painful procedures early in life having increased pain sensitivity. Putting it all together, we have reviewed two sets of very different opposing biological forces. On the left, you have the endogenous systems that alleviate pain and improve recovery. And on the right, you have the pathological processes that amplify pain. And when you think about it, the architecture of intensive farming creates a unique and highly detrimental condition where these two forces converge. On intensive farming conditions, you have no distraction, no positive social interactions, no behavior engagement, cognitive engagement, rarely rewarding experiences. So in a sense, pain is the whole world of the animal. There's no distraction away from it. At the same time, you have a number of biological processes that amplify the perception of pain, that increase the likelihood of pain from the same condition, and that prolong the duration of pain. So we call this processes pain echo chamber. So it's a situation where pain perception is promoted and healing is impaired. I hope I have convinced you that pain is also a product of place, not only affecting with environment, not only affecting the likelihood of different painful conditions, but also the intensity and duration of a same painful condition. And this has been documented in a number of cases. Take the case, for example, of salmon in recirculating aquaculture systems. Wound healing takes much, much longer in those barren intensive environments with high densities than in more natural situations. Or in our previous research in the Welfare Footprint Institute, we analyze the pain associated with gill fractures, and some of the experiments conducted in birds kept in cages pointed to very intense pain lasting 3, 4, 5 days with the bird not moving much. Pigeons, in that case, pigeons used as models of fracture pain. So the birds not moving much, flat feathers, eyes semi-closed, etc. And then you contrast that with some research groups saying that they cannot see much difference in the behavior of fractured and non-fractured egg-laying hens with gill fractures in enriched aviary environments. So I believe this is the result of differences that in environments that we discussed in this talk. So I would like to finish with 3 points. One is that current welfare assessments may be systematically underestimating intensity and duration of pain in intensive conditions if the proxy measures used to measure pain are taken from more naturalistic or enriched settings, and the other way around, they are overestimating pain in enriched settings if the indicators are taken from confinement conditions. Also, the pain echo chamber helps explain the poor validity translation of research on pain relieving drugs that are conducted with lab animals in clinical applications. And the last one is the idea that meaningful enrichment can be reframed also as a non-pharmacological form of analgesia and a promoter of healing. Thank you very much.
So thank you, Cynthia, for a really fascinating overview of an incredibly important issue, telling us that really context is everything when it comes to pain, which I think we probably sort of know as humans, and lots of us have neglected for animals. So I'll jump straight into the questions. There's one from Beata Nielsen again. Do we know how different factors interact? For instance, will distraction work more efficiently for chronically stressed subjects in barren environments?
Yes, well, thank you, Huw, and sorry I have a bit of a cough, but I think I can't recall any specific research varying the extent to which distraction would change, would have different effects based on whether animals were raised in a barren or more enriched environment. But thinking of the effect of contrast, it's possibly it would, because it would give more of a novelty to animals raised in barren environments, so it could be more effective there. Now, that said, there would be the, let's say, the constraint that animals raised in barren environments, for the various mechanisms that I reviewed, may have already some rewiring of their neural system and some heightened sensitivity to pain, and the extent to which this can be reversed is not clear. So at the same time that you would have greater contrast, on the other hand, you may have animals that are somehow limited in terms of their rewiring for pain sensitivity. So this is something that should be investigated, definitely. A very interesting question.
Yeah, I agree. Thank you. One from Suzanne Millman. Sleep may also be a natural mechanism to mitigate pain. Can there be too much stimulation in the environment? How do you balance the stimuli provoking one motivational system?
Yes, I think it's a question of balance. So definitely sleep disrupts the circadian rhythms, leads to a number of effects such as immune suppression, which also contributes to impaired healing, delayed healing, heightened sensitivity to pain. So sleep is essential. Now, that said, how much stimulation is too much stimulation? If we think of the traditional intensive farming conditions, I would say we still have a lot of room for more stimulation. Yeah, the environments are barren. There's all the issue that is being documented in recent years of boredom. So I think positive stimulation is really lacking. So it's hard to say that it would be too much by adding it. Now, that said, of course, you could have stimulation that is that leads to chronic stress, like vibration, improper lighting, too much sound, etc. That would be bad stimuli that will lead to chronic stress. So it needs to be meaningful stimulation, positive stimulation, and meaningful enrichment.
So I guess I might follow up a little bit on that. So I guess negative stressors could suppress pain perception, at least in the instance. So do you have a sense of what's worse? Is it being in a stressful environment or being in a boring environment where you can focus on your pain without distraction?
I would say if I had to, I mean, ideally none of them are like only positive environments with a meaningful enrichment. But if I had to guess based on the mechanisms that we reviewed, I would say that the barren environment is worse because in that environment there is no distraction away from the painful source. So it can be said that pain is your whole world. Distraction is very much used as a form of analgesia in poor countries. I've been working with global health for many years, and this was something that usually came up, the use of distraction as a means of analgesia, not only for acute procedures, but also for chronic conditions. So I would say the barren environment and the boredom is worse.
Yeah, I think I agree with you. There's far more questions than I can ask in the time, but I'm going to squeeze this one in from Heather Browning, who says, thank you for the fascinating and important work. Can later life experience help reverse neurophysiological changes, or is it more important to intervene early development to prevent them?
Yes, definitely early development. There is the concept of developmental programming, whereby resilience, immune competence, even the switching on and off of genes for opioid analgesia and all these mechanisms that I've mentioned, they are, let's say, established early in life. So if there was anything that we could do, I would focus on interventions at the beginning of life rather than at late life. Of course, ideally, we would have them all, but given a choice, definitely early life.
Fantastic, thank you. I'm sorry we're going to have to move on to the next talk, but there are lots more questions, and you've definitely provoked a really interesting discussion there. So thank you very much.
Thank you. Feel free to reach out to us with further questions.
Excellent, good idea. So moving on to the last talk in this session before we have a break, I'm delighted to introduce Venkatraman Manikandan, who is talking about enhancing poultry welfare AI-based vocalization analysis for stress detection and non-invasive monitoring.
Good morning, everyone. I'm Venkatraman Manikandan and I'm from Dalhousie University. I'm a master's student working under the supervision of Dr. Suresh Nithirajan. Today, I'm going to take you on a project that blends animal empathy with artificial intelligence, a system that listens to chickens and helps us understand how they feel. The idea is simple but powerful. Listening to voices and using what we hear to protect lives. Now, when we talk about AI in agriculture, we often think of automation or sensors or yield prediction. But this project is different. It's about listening, not with our ears, but with machine learning models that can recognize emotional patterns in vocalizations. Let us begin with why this matters. Chickens are the most widely farmed animals in the world. But despite their numbers, their emotions, their pain, their stress, or even contentment are rarely acknowledged. In many farms, they suffer silently. You won't see the obvious signs. Their welfare is often reduced to productivity or behavior metrics, not emotional well being. So we asked, what if their voices could tell us what their faces or bodies can't? What if sound, not sight, became the key to understanding how a chicken feels? And if we can detect those vocal cues early, it opens the door to preventive care, improving not just productivity but the animal's experience of life. Now that we have seen why emotional monitoring matters, let us look at the core challenge. It's not that chickens don't feel pain, it's that they have evolved to mask it. Their instinct pushed them to act normal even when something's wrong. Because in nature, weakness attracts predators. So even in today's control farms, a stressed or unwell bird may look like completely fine to us, on the outside. You won't see dramatic signs, no limping, no crying, just a quiet bird blending into the group. Traditional welfare checks, where someone walks through the flock and observes, are slow and dependent on human judgment. And the judgment can vary, making early signs easy to miss. So we need a better way, something faster, more consistent, and far less invasive. That's where sound comes in. Because even when chickens don't show pain with their body, they often express it through their voice, if we are paying attention. Our approach is based on a simple idea: compassion through listening. We developed a system that listens to chicken vocalizations and learns to interpret them, not just detecting noises, but recognizing emotion. Is the bird calm or stressed? Is it content or is something off? And so on. The beauty of this method is that it's non-invasive. There are no tags, no blood test, no handling. The birds aren't disturbed in any way. What happens most of the time is when you handle these birds, when you take blood samples, when you take cotton swab samples or something like that, it often induces stress to the bird. In this way, it is not happening. We simply listen and let AI do the understanding. The foundation of this system is a carefully compiled sound library of chicken vocalizations. These recordings are sourced worldwide from various data sources, and they are individually labeled single vocalizations. Each sound represents an emotional context. For example, a hen vocalizing just after laying an egg, or a pre-stress recording taken just before an environmental change. These audio clips are stored in folders based on behavioral context, such as feeding, laying, or distress. The goal is to preserve not just the sound, but the emotional context behind it. This gives a visual overview of the classical audio pipeline. In this, it starts with the chicken vocalizing, just a natural sound in the environment, something that happens all the time in the coop. The raw audio is first digitized, cleaned, and we remove the background noise. We trim silences, isolate vocal events. This pre-processing ensures that only meaningful sounds reach the model. Then the signal is fed to a custom code base. This is where we perform feature extraction. You can see examples on the right. MFCCs which simulate how human ear hears. They have been used for years in speech recognitions. Spectrograms which show how sound energy shifts over time and frequency. And statistical features like zero crossing rate, RMS, centroid, spectral contrast, etc. Each of these captures a different fingerprint of the sound. All these features form a structured input for the traditional machine learning models. And even though this isn't deep learning yet, it was enough to show us that emotional cues really do exist in these vocal patterns, even when they are subtle or masked by background noises. This is what a chicken's voice looks like when visualized. What you're seeing here is a spectrogram. It maps sound over time and frequency. Each horizontal line represents a frequency, and each vertical slice represents a moment in time. The color shows how strong that sound is, with brighter bands meaning louder or more intense energy. Spectrograms help us distinguish between different types of calls. For example, a calm cluck might produce soft, low frequency waves with gentle slopes, while distress calls are sharper, more jagged, and typically shifts to the higher frequency. Even if our ears don't catch the difference, these patterns become visually obvious. Every vocalization, whether it is curiosity or stress or contentment, creates a unique acoustic fingerprint. And once we learn how to read these patterns, we can start training our machines to recognize them too, like teaching a musical ear to an algorithm. This becomes the foundation for an automated emotion detection, not just by sound, but by the shape of the sound. This This shows the complete pipeline from raw sound to welfare decision. It begins with audio recording. Microphones placed in the environment capture the sounds of the chickens. Next come pre-processing. This is where I told you, we remove the background noise, audio is trimmed, and isolate meaningful vocalizations. Then we perform feature extractions, pulling out even elements like frequency, energy, and the texture of the call. The features are passed through scaling to normalize the values and prepare them for learning. The processed data is then sent to classifier models. These are like machine learning algorithms trained to detect these patterns linked to the emotional states of the animals. Finally, the system makes welfare decision. Is the bird showing signs of health, stress, unhealthy, illness, just based on the sound it makes. Once we could classify calls into stress, calm, we realized something deeper. Chickens weren't just making sound, they were communicating. These clucks, these coos, or these quacks carry context, like a hen calling out after laying or a chick seeking its mother. To truly understand welfare, we need to move beyond a yes or no classification. We needed models that could interpret these sounds more like a language, like our English language, capturing not just the tone or energy, but the rhythm, the pauses, and the emotional shifts. This meant shifting from sound detection to semantic understanding, where the system starts listening like a human listener would. This is where the AI gets interesting. Now let me walk you through how wave to wake works. This is the part of the model that listens. You can think of a baby that learns to understand speech. It doesn't know words yet, but it listens to tones, the patterns, the rhythms, and slowly starts making sense of the meanings. Wave to wake does something very similar. It takes the raw chicken vocalizations, just the waveform, and breaks it into small sound slices. Then during the training, it hides some of these slices and tries to guess what's missing, which forces it to learn how chicken sounds when they are stressed, calm, or communicating. It does not rely on handcrafted features like MFCCs. Instead, it builds its own vocabulary of sound tokens. These are the learned patterns that repeat in the chicken calls. And the tokens that you will see in the next slide, they are called the vocabularies Wave to Wake had created. They are not words, but they act like words. The model has learned which types of clucks and calls show up together and what they mean emotionally. Now that we have these sound tokens from wave to wake, the next step is to understand what they mean. And that's where BERT comes in. BERT is a transformer model that treats the sound tokens like a sentence. It looks at the order, the spacing, and the patterns in these tokens. Just like how we hear emotion in someone's voice by how they speak, not just by what they are saying, we also understand their emotions from the way they speak. BERT adds context. It knows which cluck came first, where there was a pause, or when the tone suddenly changed. It uses attention mechanism to highlight the most important moments in a vocal sequence. Layer by layer, it builds an understanding. Was the call urgent? Was it confused? What was the bird trying to say? Was it trying to rejoin others? Etc. Eventually, the model gives us a final interpretation, not just a label, unlike the classification, but a mood. It might say the bird is distressed or that the call sounds are calm and relaxed and the chicken is looking for group, etc. So instead of hearing sound, the system is now understanding communication. That's the power of semantic learning, from meaningless clucks to meaningful moods. Bird does not hear like a sensor, it listens like a sensor. So, what did we actually hear through all of this? Calm chicks tend to sound soft and rhythmic, a kind of steady, familiar tone. But when chickens are stressed, their calls change. They become short, sharp, and harsh. Even after a mild stress event, the tone shifts noticeably. The amazing thing is that these changes happen before any visible signs are shown up. The voice becomes kind of an early warning system, reflecting emotional changes in real time. Just like humans, chickens carry emotions in their voice, and with the right tools, we can detect it. This approach is just clever. It is practical. It is non-invasive. No need to tag or handle the birds. The chickens don't know that even they are being monitored, which means no added stress. It works across entire flocks, not just few birds, giving a full picture of well-being. Instead of scattered snapshots, because it detects early emotional changes, farmers can respond faster with better nutrition, better care, and even better mental health outcomes for the bird. And the setup is simple. The farmer doesn't have to think hard. He doesn't know how to have degrees in computer science or something like that. It's just microphones and smart models. At the end of the day, this helps both the chicken and the farmers. Better welfare leads to better outcomes across the board. So to end, here's the takeaway. Sound is very powerful. It can tell us when an animal is stressed, curious, or in distress, long before we see it in our eyes. By listening intelligently with compassion and AI, we can support better welfare, better farming, and a better future for poultry. If you'd like to explore the technical details, feel free to scan any of the QR codes here. They'll take you to our papers. If you'd like to connect, whether for collaboration, discussion, or just stay in touch, you can directly scan the QR here that leads to my LinkedIn or email me directly. Thank you all for listening and being a part of this movement towards animal-centered innovation. Thank you.
Thank you very much, Venkatraman. Welcome, and thank you for sharing that really interesting work, which I commend, looking for non-invasive ways to monitor welfare, which is clearly incredibly needed. There's a question from my colleague Beata Nielsen, which is similar to one that I was going to ask. So she says, perhaps I missed it, but can you briefly explain how you validated the link between the call and the state of the animal? So I guess, in other words, she's worrying that there might be a bit of a circular argument here. If you assume that the call is related to negative welfare, then it will be classified as such.
So our data set is basically annotated, which means based on experts and based on the environmental conditions, we recorded voices from the animal. Say, for example, I would give you an example, a very, very expressive example. So in one setup, what we did was the animal was placed a visual stimuli to induce a stress was given. For example, an umbrella was suddenly opened in front of them. So that kind of agitates them or creates an anxiety, creates a stress for them. So after that stress was created, we recorded the voices. So we recorded the voice before the stress and we recorded after the stress was induced. Another example is we stimulated to give an auditory stress, not just visual stress, to give an auditory stimuli. What we did was we stimulated dog barking sounds. Like we made sounds that were similar to dog barking, and we made the chickens agitated, and from that we recorded the sound. So this was related to the stress part, and from experts and other the subject matter people, we were able to identify sounds. Like we classified sounds based on like if they had laid eggs, what type of sound they make, and if they say they have fight between another hen or something, what sound they make. So we recorded those sounds and we used those sounds to our data set. And another similar example I can give is another data set that we used is where we recorded the voices of animals that of the hens that had a respiratory disease and which did not have a respiratory disease. Like it's more of a healthy animal, unhealthy animal. So from that, we were able to distinguish these calls.
Great, thank you. I've got a question from Claire Witham, which is on a sort of a practical note. She wonders how well it could cope in a large chicken shed where there is lots of noise. In this case, is your model going to be capable of telling anything about individual welfare, or does it only work at a group level?
So that is where our future work is being done. So currently the model works, it works in a research environment. So we have to test it out in a lab, in a commercial environment where there is more of noises from maybe metal banging sounds or something similar. So industrial noises will be there. So that is where the noise isolation comes, the subtraction of noise from the animal sound comes. That is the part that's the future we are heading towards. That's the future direction of our research work. So that is, we are planning to tackle that using noise suppression algorithms that can help us to reduce these industrial noises and just extract the animal noise. I mean animal sound. Yeah.
Okay, thanks. One from Suzanne Millman here. She says this is a very exciting development and she likes your approach. Can the tool quantify sequences and durations of calls to help us objectively assess stress recovery and resilience?
Yes. So that is an interesting insight actually. The wave to wake that we use is actually separates the sound into chunks, and from these chunks we can actually track on how or at what time sequence specifically the stress or the event happened, or what the animal is trying to say at that particular sequence. We have in our paper also we have explained. I did not include that in the presentation due to time constraints. So we have also added a time series analysis in our paper, which analyzes like how the voice changes over a period of time, and we have also analyzed how it changes over the age as the animal matures from day 0 to maybe 5 weeks, 6 weeks. So how the voice changes to the responses or the stimuli, we have also analyzed that as well. So we have done a time series analysis, but I was not able to include in this due to the time constraints, but it is there in our paper as well.
Okay, thank you very much. And I guess related to some of this, I'm going to collapse a few questions together, but people are basically asking, is this still in the trial stage or is it in commercial use, or is the software available if people want to try it?
So we are still in a research facility based environment. We are not yet commercialized, but our plan is to commercialize. As one of the earlier questions I remember asking regarding in the commercial environment, there would be noises and other things. So that is the part we are working on. So in order to commercialize this, we need more stronger noise reduction algorithms, because in an industrial setting, there will be a lot of noises. I visited a couple of farms around here, Nova Scotia and New Brunswick. So there were a lot of other noises than chicken calls. So we need a stronger noise reduction algorithm. That's where we are working on. And once we are done that, it would be commercialized. It can be commercialized, yes.
Okay, brilliant. So I guess people will be looking forward to trying it in the real world. So thank you very much. It's really exciting to hear about these innovations. Unfortunately, we're a bit pressed for time, so there are lots of other questions, but we'll tackle them later if we can. So thank you very much for your talk. Thank you to all the presenters in this session. We're going to take a short break now until 3:10 British summer time. So about 3:10 British summer time, and we'll come back. So what we'll do is we'll close this link and reopen another session, because that gives us time to digitize the videos. So if you click on the link that you clicked on in your emails to come back at 3:10, we'll see you for the next session. And thank you everyone for attending and for excellent talks to start off the conference. Thanks everyone.